Hey, what's up, you guys? Yes, we're back for another episode of Biology Unlimited. I'm Paola.
And I'm Sophia.
Today we are here to teach you about genetics.
We will teach you the background of modern genetics, and at the end, we will include a step-by-step tutorial on how to draw a Punnett square.
Exciting stuff. After this video, you all will practically be Gregor Mendel himself.
Let's dive in. Everything we know about genetics today stems from a monk named Gregor Mendel. He lived in the mid-1800s in an abbey garden and documented inheritance in pea plants.
He did lots of experiments on pea plants by cross-pollinating these pea plants with opposite traits such as tall and short and white and purple. By meticulously observing the pea plant and keeping detailed notes, Mendel was eventually able to establish the first ever rules of inheritance.
Mendel summarized his findings into 3 laws: the law of segregation, the law of independent assortment, and the law of dominance.
The law of segregation states that each allele segregates into separate gametes. During meiosis, the alleles segregate. Each allele is then packaged into a separate gamete. Then, during fertilization, one gamete from each parent comes together to form a zygote.
The law of independent assortment states that different genes separate into gametes independently. This means that traits are transmitted to offspring independently of one another.
The law of dominance states that dominant genes will be expressed over recessive genes.
Mendel used test crosses to determine the genotype of an offspring between 2 parents. He used Punnett squares to perform test crosses.
Hold on, Sophia. Our viewers might need a little more information. It's time for a little vocabulary lesson. Homozygous means that a genotype has the same alleles. You can have 2 different types of homozygous genotypes: homozygous dominant or homozygous recessive. Homozygous dominant has 2 copies of the dominant allele, and homozygous recessive has 2 copies of the recessive allele. Heterozygous means that a genotype has different alleles.
Thanks, Paola. So, to test cross, you first need to draw a box with 4 squares, called a Punnett square. Next, you need to determine each parent's genotype. Choose a letter to represent each allele. For our example, we will be using big A, which is dominant, for purple scales, and little a, which is recessive, for blue scales.
Next, determine the genotype of each parent. For our example, we will be crossing a homozygous dominant parent with a heterozygous parent. Label the rows with one parent's genotype and label the columns with the other parent's genotype.
Hold up. The process of separating the alleles relates to the law of segregation. Remember that from earlier?
Oh yeah, thanks for reminding me.
The rest of the Punnett square is easy. For each box, look to the allele in the corresponding row and the allele in the corresponding column. Write both these letters in the empty box. This cross is a possible genotype of the offspring. Now, do this for the rest of the boxes.
Dominance: if any of your pairs have a big A, then purple scales will be expressed. The only way the offspring can have blue scales is if there's 2 small A's.
So now we will analyze the results of our test cross. 2 out of 4 squares are homozygous dominant, which means that there is a 50% chance of having offspring that are homozygous dominant. 2 out of 4 squares are heterozygous, which means that there is a 50% chance of having offspring that is heterozygous. However, all of these crosses have a dominant A, which means that all of the crosses will have purple scales. There is no possibility of a recessive cross, so no offspring could have blue scales.
Thanks for tuning in to another episode of Biology Unlimited. We hope you learned a lot. Like and subscribe down below.